System, we have the cerebrum. Okay, so we have 3 slides for the cerebrum. This one is Asian E. Okay, so as indicated by the stain. Okay. So what we have to identify here would be the layers of the cerebrum. So here we have the gray matter, okay. And then the middle portion would be the white matter. So the meninge that we would identify here would be the pia. It's the innermost meninge. And then in the gray matter, the outermost layer, we have the molecular. And then we have here the 5 layers, okay, which is a little bit difficult to distinguish. Okay, so we have here the molecular, we have the presence of neuroglial cells. In this portion, you would identify the pyramidal cells, small pyramidal cells, and they become bigger. And then you have the larger pyramidal cells. This one is already in the white matter. The other slide would be better. This is silver impregnation. It has a different appearance, so we just look at the area we're in. We would see some differentiation. So sometimes the meninge would be merged as one. In this case, we have the pia and we have the arachnoid being separate. The dura is thicker. So here you have already the white matter, and this would be the gray matter. And the outermost area will be the molecular. You will see the neuroglial cells. And then the next layer would be this would be the small pyramidal, or the external granular. You can see small pyramidal cells. The third layer would be the external pyramidal or outer pyramidal, or we would see the presence of pyramidal cells, but sometimes it's indistinguishable from the second layer. Then the fourth layer, you can see less of the pyramidal cells, more of the granule cells. That's the internal granular. And then this one would be the layer of the internal pyramidal, wherein we have large pyramidal cells. And then here in this portion would be the fusiform, wherein we are very near the white matter. Here you see the meninges again.
Specific meninges.
Specific. For this part, this would be the pia and arachnoid.
Yan parang dark shadow daw.
Better picture with regards to the meninges in the spinal cord. if ever I'm going to give you the meninges more in this kind. So this would be the molecular, and then this would be, these are the pyramidal and granular layer. So a little bit difficult to identify with this particular stain. Okay? It primarily focuses on the appearances. Okay? So you have large cells as the pyramidal cells, but it's difficult to identify the granule cells. Smaller round cells, they will be seen in the internal granular. Okay, next we have the cerebellum. So the cerebellum would be thrown into several folds. The convoluted folds would be known as the cerebellar folia, and then they are separated by the sulcus, interfolial sulcus. Okay. So these are convoluted folds of folia. So the one in the middle represent the white matter. This layer would be composed of the gray matter. Again, the gray matter, up to this portion, we have this less cellular area called the molecular layer. You have the Purkinje cell layer, and then you have the granular layer.
Granular.
So that is in the molecular. So in the molecular, you would see the basket cells. So these are the cells, the basket cells. And then you have the Purkinje cell, which they have dendrites that would proceed into the molecular layer. And then you have the granular layer containing small round cells called the granule cells. And then you have spaces, pink spaces there, which would be called the glomeruli, containing the synaptic processes or complexes. You can see occasional larger cells with vesicular nucleus. This would be the Golgi type 2 cells. And then here you have the white matter containing the myelinated axons as well as the glial cells. So referring to the supporting cells. There's another slide. This is the silver stain. So the silver stain is used to highlight the dendritic processes as well as the presence of the Purkinje. Purkinje. Okay, so you have the dendritic processes. So this would be in the area of the molecular layer, Purkinje, dendritic processes. Next we go to spinal cord. So we have a limited view for the scanning because it's a big structure. So kindly try to look at the entire area of the outer portions, the ranges. So this would be the dura. It's a large connective layer. Okay? And then the space would be the subdural space. Underneath, you have portions that can be separated into the arachnoid, subarachnoid, then you have the pia. Okay? But there are some portions to which they are seen as one, okay? Called the pia arachnoid. The majority of the areas for the spinal cord would consist of the white matter, the one in the middle, and the gray matter. So the center of it all would be the central canal. Okay? So it would be lined by ependymuscles. So this would represent the central commissure, okay, which would link the central canal to the horns. So the upper portion would be the dorsal, and you have the ventral. Or the anterior and the posterior. So located within the horns, we have the motor neurons. The large cells. It's better seen in the microscope, okay? Especially its nucleus. This one is, you have the big cell body. I think this one is the nucleus. And then you have the nucleolus, the one in the middle. Okay? So differentiating feature between the dendrite and the axon, there's the presence of missile bodies into the, I'm not sure if this one is the axon and this one is the dendrite. Okay? So if there's a presence of missile bodies into that particular process, that's the dendrite. And then you have a lot of supporting cells, the glial cells. In the white matter, we would see the myelinated axons. Okay, so these are myelinated axons. The one in the middle is the axon. And then this would be the myelin. And then you have the myelin sheath. So myelin sheath, myelin, the space in the middle, at the space, and then you have the axon. So we go to the spinal ganglion, a sympathetic nerve ganglion. For the sympathetic nerve ganglion, we look into the ganglion cells. So these are large cells. We have a large cell, round cell body. Ganglion cells. They possess a prominent nucleolus, nucleus, okay, vesicular nucleus, the prominence of the nucleoli. Okay. And then they are surrounded by small cells called the satellite cells. And notice there are fibers running along with it. So these are the axonal fibers. Here's another picture of spinal ganglion, but this time it's both are seen in longitudinal section. So they can be identified easily by their plump cell body. The prominence of the nucleolus. Okay, next we go to the nerve bundles. For the nerve bundles, let's start first with the coverings. So we have connective tissue covering for the nerves. Okay, so individual fascicles, they're surrounded by the perineurium. So if we have the presence of a connective tissue layer surrounding the two fascicles, that would be the epineurium. Okay, but as you can see here, it would be composed of adipose tissue. So what we will see would be the perineurium. And then you have the cross section of the axons. Okay, again, the axons would be located in the center, surrounded by myelin, and then you have the myelin sheath. So some of the slides here are redundant. Okay. So you can see similar appearances. This would be the optic nerve accompanied by the vascular channels. Again, you have only the perineurium, and then you have the presence of the axons. Okay. I think these are Schwann cells. So here you have the axons, and then you have the myelin within. So look into the longitudinal section of look into the longitudinal section of the nerve. So here you have one nerve. Okay, you have the myelin, you have the axon, myelin nerve sheath. This one would be the axon nerve sheath. Axon nerve sheath. Okay, so you can see.
What's the work?
Sometimes you won't be able to identify readily the nodes of Ranvier. So we can see it in some other things. This is the silver stain. This one, those are the nodes of Ranvier.
Intercalated disc.
Okay. So myelinated, so myelin sheath, axon, nodes of Ranvier. So you can see easily in some things. The neurovascular bundle, see. This one has an epineurium. Okay. So the epineurium would be it contains connective tissue surrounding clusters of nerve fascicles. Okay. And then you have the perineurium. So this is motor end plate. What are these structures? So we look into them. So these are skeletal muscle, striations. So these are the axons.
Oh.
And then these are the motor entry.
No, eh.
So they would attach, okay, they would be attached to the individual skeletal muscles.
Wow.
So for voluntary action. Then we go to some receptors that will be present in the body. This would be the Pacinian corpuscle. So this is not in the skin. Okay? So this one would be pancreas. So this is the pancreas highlighted by the presence of. So those are islet. Islet of Langerhans. Islet of Langerhans. So that the serous acini are slightly obscured because of the compactness of the tissue. This is what we are looking for. This is the Pacinian corpuscle. So it's a receptor that is involved in vibration and pressure sense. So you have lamellae of connective tissue, fibroblasts. Then the middle one, this is the axon. That's the axon. This one is a nerve fiber. So it's involved in vibration and pressure sense. And then the last slide is Meissner's corpuscle. So the Meissner's corpuscle is a receptor for light touch. So they are seen in the fingers. So where do we look for them? We have the epidermis, the dermis. You call this as the retipex, and we have the dermal papillae. So you have the epidermis, retipex, dermal papillae. So this is the Meissner's corpuscle. So epidermis, retipex, dermis, dermal papillae. So it's located in the dermal papillae, composed of Schwann cells and nerve fibers. Okay, so it's involved in light touch. Again. So that's it. Okay, we look for that one. Okay, so looking at your slides.